we want to exploit the signaling cascade by blocking with the 5,7-dihydroxytryptophan. Okay. So we provide a different concentration of 5,7-dihydroxytryptophan, and we allow them to learn to the novel odor. Novel odor in the sense the chopped apple with the cinnamon. Okay. So basically we classify them into three different categories as like an outfly, for example, just flying around. Because we want to make sure that once we made a drill into the skull and deliver this 5,7-dihydroxytryptophan, it will not affect the normal physiology and the behavior of the animal. So the outfly is a very essential one. We want to make sure that it's like that, flying perfectly. So that's fine. They are flying perfectly. There is not much difference. And then the attempt towards the food, and they responding to the food like a feeding mode. So in these two cases, the one with the higher concentration of 5,7-dihydroxytryptophan one, they showed very poor learning, and also during the memory test, the response is very good. So this provide an idea. Yes, the depletion of a 5-HT, it changed the behavior of the bats, and subsequently you want to confirm whether the serotonin level is like altered or not by the treatment of 5,7-dihydroxytryptophan. So in the olfactory bulb, okay, the higher concentration treated one is goes down, but not in the forebrain and hindbrain. Okay. So it's very site specific. It's not transported to the other region of the brain. It's only depleted in an olfactory bulb. That's what the outfly and everything is normal. Okay. And similarly, we want to see that, because in the transcriptome I show that once it is released into the GABA junction, it may activate the different receptors. So we pick up the 5-HT1A receptor. So the expression of 5-HT1A receptor is much less compared to that control or saline treated one. Subsequently, in the signaling scheme, I showed that the extracellular kinase phosphorylation is very essential to translocate into the nucleus and to activate the universal element, the cytoplasmic response element. Okay. So when we examined the northern plot, you may remember that from mRNA to DNA analysis. So the mRNA level of ERK1 is really goes down when the level of serotonin goes down. Okay. So even though there is a stimulus, the cellular signaling cascade got not activated. So the level of mRNA of ERK1 and CREB1 goes down. So the same got reflected in a phosphorylation event. Here, the ERK2 and CREB phosphorylations is less compared to the stimulated one with the. So this given we move one stage forward into the nucleus, and the next stage is like in a signaling cascade. You want to confirm the early response gene. Okay. So this early response gene is very specific to activate to the odor, or it's like involving with a different business in the olfactory bulb. Okay. So here we examine the immediate early response gene like EGR1. Okay. So this is like a saline based on with the olfactory stimulus, and with the other one, the 5,7-dioxygenated, the serotonin depleted. When the level of serotonin depleted, the expression of EGR1 is very less. And then we want to move the next stage, because we never know that how long the expression will be in a EGR. So based on the time of expression, we want to deliver the drug. Okay? So we systematically analyze after the stimulus how long the expression will stay. Okay? So the stage starts from the 0 minute and the highest reach to the 60 minute, and then 2 hours and 6 hours is back to the basal. So it's a kind of transit expression to the stimulus. It goes to the higher level in an hour, and then it goes to the basal. So now we know that in what time we have to sacrifice the animal after the treatment or the stimulus to examine the study. Okay. So based on this, we design an antisense oligonucleotide. You studied about an RNAi or siRNA. Okay. So this siRNA or the RNAi directly interfere with the transcription. So here with the RNAi, there is like an advantage with the study in a cell line, but in the animal model, the stability of RNA may not be very long. So we decided to use an antisense DNA. Okay. So this DNA, short DNA molecule, it binds with an active mRNA and it will not allow to transcript. Okay. So we designed an antisense oligonucleotide targeting this EGR1 expression, and as a control we use a nonsense one. Nonsense one is like a scrambled form of oligonucleotide, but the same combinations of antisense, but the arrangement is different. So it will present there, but it will not bind with the mRNA. Okay. So we provide this antisense oligonucleotide directly injected into the olfactory bulb, but now the level of serotonin is fine. Okay. So there is a stimulus, serotonin level is fine, and we block the transcription of EGR1. We tested for 1 hour before the olfactory training, and then we tested the memory after 24 hours. Okay. So what will happen when we provide like a 50 picomole of antisense oligonucleotide? They learned very poorly and they responded very poorly to the memory test. Okay. So this is the way we examine the expression of an EGR1 after the antisense oligonucleotide treatment. So this is like an antisense and the nonsense, the scrambled form and something. So the designed EGR1 antisense oligonucleotide effectively block the transcription, and the level of protein is very less. But in the nonsense one and the controlled one, it's perfectly increasing based on our previous experiment. Okay. So this is an early response gene, and it just move on to the late response. The late response genes or the protein, they located in the synaptic region. Okay. So these expressions make an enlargement of the synapses. Okay. So when there is an enlargement of the synapses, the gap, the synaptic gap will be less. Okay. So when the gap is very less, there will be an interaction of a protein from pre-synaptic to the post-synaptic, so that the communication from one neuron to another neuron. So once the stimulus is over, the enlargement will go down and the gap will increase, the communication will stop. Okay. So in this one, there are like a major protein which involved in the enlargement is like a post-synaptic density protein. Okay, PSD 95. So there are many post-synaptic density protein. Among them, 95 is that high